Hi everyone. Today I'm going to talk on the topic immunity. You all know that the immunity is most important aspect to get protection from the various microorganisms or toxic products for which human beings are not only human beings, any living animal is going to be protected from the microorganisms. So this particular caricature will depict how the immunity will protect an individual like in a big wall or barrier. So in this particular topic, we are going to learn about basics and its applications of immunity. So learning objectives are historical considerations of immunity, definition types of immunity, factors influencing degree of innate immunity and its mechanisms, acquired immunity and its mechanisms and its applications, and other important aspects of acquired immunity in the public health will be learned from this particular topic. So historical considerations, if you see, the immunity is the word derived from immunitas, which is a Latin word, which gives the meaning that the protection are free from the disease. Chinese customs, so without knowing the concepts of immunity, there was a custom in the China, that is inhalation of powder made from smallpox lesions to get protection from the disease of smallpox. And in Athens, 5th century before Christ, the people who wants to treat individuals who are suffering with the plague, they should get recovery from the plague disease. And 19th century and the Christ era, the material from cowpox was obtained and prepared to give the protection for the smallpox. This is the work which everybody knows, done by Edward Jenner. And Emil von Behring and Kitasato in 1890 described the mechanisms of immunity and how the resistance or immunity would be transformed by serum through the antibodies. So, if you see the immunity can be classified into innate immunity and acquired immunity. And again, acquired immunity would be divided into active and passive immunity. Before going for that, we have to know what is the definition for immunity. It's a state of resistance or insusceptibility exhibited by the host towards the microorganisms, toxic molecules, or the foreign cells. So this kind of immunity may be inborn immunity or it may be acquired during the lifetime of an individual. So this acquired immunity might be active or passive. And again, in case of active acquired immunity, the host immune system has to work effectively to produce antibodies on exposure to the antigen either naturally or by means of artificial way. And in case of passive acquired immunity, the antibodies which are prepared in the other host would be transformed either manually or naturally to the other host to produce the resistance. About each and every one, we will discuss in the next few slides. And first thing is about innate immunity. This is the quote quoted by the Hippocrates. The natural forces within us are the true healers of the disease. So before knowing the concept of innate immunity, he mentioned that the natural forces are there in our body which are responsible for getting resistance against many number of diseases. And if you see the innate immunity, by definition, it's an inborn resistance against the microbial agents that an individual possesses by birth due to genetic or environmental makeup. That means individual will have the resistance by means of structure or by means of certain cells in the body and gives the protection against the microorganisms for which the human being is getting exposed. And if you see the properties of this innate immunity, this acts in minutes That means as soon as the microbial agent entered the body, the innate immunity starts working, and the prior microbial exposure is not at all required to produce this kind of immunity, and acts against limited number of antigens only. It's non-specific, which can try to attack each and every type of antigen entered the body, and there won't be any memory, that's immunological memory, followed by exposure to an antigen. And it may be confined to the species level or races level or the individual levels. So if you see the species specific innate immunity, and innate immunity towards a microbe by all members of given species. For example, if you take the Bacillus anthracis, which you know it's a very important pathogen, including human beings, frogs are resistant for this Bacillus anthracis, whereas toads are susceptible to this particular Bacillus anthracis, which can produce anthrax even in the toads. So even though the both are comes under the same family, frogs and toads, certain species of the frogs are amphibians. Sorry, the amphibians are resistant. Certain species of amphibians are sensitive. And racial specific, innate immunity confined to a particular race by a particular microbe. You all know that there are so many races are there in the world, and even though all of us are human beings, we have Negroes, we have Mongolians, we have Egyptians, and we had Caucasoids or whites, like different races are there. So the natural immunity confined to a particular race by a particular microbe. So, for example, if you take the tuberculosis, Negroes are resistant to tuberculosis, whereas whites, including us, Caucasoids, are sensitive. That means we may suffer with this tuberculosis disease, whereas Negroes may not. And anthrax is a disease of animals, primarily we all know. But Algerian sheep, even though it's animal, that animal is resistant to the anthrax disease. And malaria is a disease of human beings, but Africans are resistant to falciparum malaria. How this is happening? This is because of vaccination? No, not at all. That's because of innate immunity, which is there in the body, already existing in the body, and giving protection depending on different types of races for the same organism. And individual specific innate immunity means antimicrobial resistance confined to a particular individual by microbe, not for others. Say, for example, if you are, if the two people are there, two children are there, two parents, even though both of those children comes from the same parenthood, but still one baby may have the resistance or susceptibility against the microorganisms in 1 degree, and another baby may have the either resistance or susceptibility towards the microorganisms in the form of other degree. So the susceptibility pattern or resistance pattern varies from individual to individual. That is known as individual specific innate immunity. And what are the factors influencing this innate immunity? There are four important factors are there. One is the nutrition, exercise, breathing, meditation. So even today, these things will work to build up very good immunity in the host. And which are the factors determining the degree of innate immunity? The first one to be discussed in this list is age. If you see the age, the rubella is common in fetal life because of the immature immune system, and chickenpox and measles are common in children, and UTI is common in adults. So as the age advances, the immunological functions will goes on waning, and they goes on decreasing. So the adults are more prone for varieties of infections when compared to the children. And the hormones also are having a role on the degree of innate immunity. Disorders like diabetes mellitus, where the insulin deficiency is seen, these people are more prone for certain infections like staphylococcal abscesses are common among these people. Lack of sex hormones before puberty make the individuals to prone for gonococcal and dermatophytic infections, especially Microsporum adoni. Microsporum adoni is a dermatophyte which causes for the scalp infections among the children. And automatic recovery or a spontaneous recovery is possible, followed by the onset of puberty. And adrenal dysfunction, if corticosteroids which are produced by the adrenal cortex, either increased levels of corticosteroids will cause us for the immune suppression, and the decreased levels the increased levels are, causes for the anti-inflammatory activity, and decreased levels is responsible for the disease production even by the commensal organisms in the body. So the normal levels have to of the corticosteroids have to be maintained to maintain the proper immunity. And another important hormone is the thyroid hormone. It also has got a great role in the immunological functions, because those thyroid hormones are having great role in the metabolism of so many things in the body. Next one is nutrition. Malnutrition, especially protein energy malnutrition, is responsible for decreased antibody production, and which results in decreased immunity. And the mechanisms of innate immunity, there are seven mechanisms are there. The first one among them is the interaction between the receptors. What are those receptors? That's a question now. The microbial surface molecules, named as microbes associated molecular patterns, MAMPs, these are all peptidoglycan on the cell wall, and lipopolysaccharide, teichoic acid, and lipoprotein, which are present on the cell wall of the bacterium, is one type of receptors. And another type of receptors which are present on host cells are pattern recognition receptors, which are popularly known as toll-like receptors. These toll-like receptors present on the surface of the phagocytes, which can recognize microbes associated molecular patterns like peptidoglycan, LPS, teichoic acid, and lipoprotein. So, whenever the microbe enter into the body, the reaction or interaction will occur between these two receptors and responsible for initiation of immunological responses. So, if you see the binding between the MAMPs and toll-like receptors activate transcription factors which stimulate the expression of genes coding for the production of cytokines and enzymes which are responsible for innate immunity. And here you can see the toll-like receptors. This is the below one is the macrophage, and the macrophage will express the toll-like receptors. And gram-negative bacteria, which are having the polysaccharide antigen, and capsular polysaccharide antigen, and lipopolysaccharide. And the same way, the even the gram positive organisms also will have. So the reaction between the interaction between the receptors which are present in the macrophage and either gram negative or gram positive bacteria will activate the transcription factors like nuclear factors, which promotes the production of cytokines. And another transcription factor activated by this reaction is interferon regulating regulatory factors, which can stimulate the production of interferon alpha and beta. All these are responsible for the expression of immunological reactions against a microbial agent. Now, the second one is anatomical barriers, which are responsible for innate immunity. These anatomical structures are present in the body for some other purpose, but they also participate in natural immunity. The first one is the skin. The skin mechanically prevents the entry of microbes and produces large quantities of sebum, which consists of peptides and fatty acids, which acts on the microorganisms to kill them. And killing of microbes by intraepithelial lymphocytes which are present, the lymphocytes present in the intraepithelial cells, also part of the innate immunity. Mucosal barriers like mucous membranes, which prevents the entry of microbes by producing the mucus, and this mucus entraps the many number of microbes and propel them out to the ciliary function of mucosal membranes. Cilia present in the lower respiratory tract also remove the microbial agents from the mucosal surfaces. And another physiological barriers of innate immunity are the temperature. Normal body temperature inhibits the growth of many number of bacteria. The body temperature around 37, whereas these microorganisms will grow best at 27 or at the 30 degrees centigrade. And gastric pH, gastric acidity inhibits most of the microorganisms. And secretory products of mucosa like saliva. This saliva consists of certain enzymes which can damage the bacteria by damaging the cell wall and cell membrane. TS consists of lysozyme enzyme that destroys the peptidoglycan layer of bacterial cell wall, thereby it can inhibit the entry of microorganisms. Gastric juice, the hydrochloric acid present in the gastric juice, which is unfavorable for the growth of microorganisms. Trypsin hydrolyzes the bacterial proteins and thereby cause for destruction of the bacteria. And the bile salts, which are present in the body, interface with the bacterial cell membrane. Fatty acids denature the bacterial proteins. Spermine, which is present in the semen, inhibits the growth of gram-positive bacteria, and acidic pH of vagina also won't allow the microorganisms to establish their infection in the vagina. And lactoferrin binds to iron, thus interferes with the acquisition of iron by the bacteria. Certain bacteria to grow, the iron is must. So the lactoferrin, which is present in the body, will bind or chelate the iron so that those particular pathogens can't grow. Like this, varieties of things like the secretions from the mucosa, body temperature, and pH also will inhibit the microorganisms to enter and to produce the disease, which are all naturally present in the body and participates in the natural immunity. And another fourth one is antibacterial or the antiviral substances in the blood and tissues. These substances which are present in the tissues and can kill the bacteria are the important structures of those microbial agents. First one is beta lysine, which acts against the capsule of the bacilli, and certain bacilli, the capsules are the main pathogenic parts of the bacteria. So once the capsules are inhibited or destroyed by this beta lysine, the microorganisms can't initiate the inflammatory process. Polypeptides are produced from the leukocytes, also from the platelets by means of which are known as leukins and plakins, which are also having the antibacterial or antiviral agents. Defensins like alpha and beta defensins are present, which makes the pores in the bacterial membrane and causing for the destruction of the bacteria. Lactic acid present in the muscle tissues and also produced in the inflammatory zone, and this acids acts on the microbes to kill them. Interferons produced by the virally infected cells in the body, and these interferons acts on the viruses to kill them. That's why most of the times the viral infections are self-limiting diseases. Humoral factors like piperidin and complement also acts like antibacterial and antiviral substances. And some of the cellular components also participate responsible for innate immunity. The first one is the basophils and mast cells. These cells releases the chemicals that mediate inflammation and allergic response. And another thing is the neutrophils, these are also known as microphages, which can ingest and destroy the uh, the invader or microbial agent which invades the body cells or organs. Eosinophils destroy the antibody coated invaders. So the eosinophils are again granulocytes which destroys the microbes which are coated by antibody. And macrophages and monocytes, we all know that these are the major cells of phagocytosis present in epithelial lining of mucosa and responsible for the phagocytosis, and they're also responsible for the presentation of the antigen to the T lymphocytes to for the further processing of the antigen. Dendritic cells recognizes the microbes in skin and mucosal surfaces, and they also act like antigen presenters or antigen presenting cells to the T lymphocytes to kill them by the T lymphocyte activity. And NK cells recognize and destroy cancer cells and also the virally infected cells. And like this, the varieties of cells like basophils and mast cells, neutrophils, eosinophils, macrophages, monocytes, dendritic cells, NK cells, all are present in the body naturally. And by means of all these activities, primary functions, they can inhibit the microorganisms to establish in the body to produce the infectious disease. The complement pathways also, so there are two important complement pathways are there. One is classical alternate pathway and next one is mannose binding pathway. So the alternate pathway activated by the endotoxins. Endotoxins, you all know that produced by the gram-negative bacteria. They can activate the alternative pathway, and mannose binding pathway activated by the mannose residues which are present on the bacterial surface. So the either by the endotoxins produced by the gram-negative bacteria or the mannose residues present again most of the times on gram-negative bacterial surfaces can activate these complement pathways and leads to lysis of the targeted cells and also stimulates the inflammation and also stimulates the acquired immunity. And how it's responsible for acquired immunity, we can understand later while we're talking about the separate chapter that's complement is a separate chapter you have in immunity. While discussing about it, we can learn how this is responsible for acquired immunity. Now, this inflammatory process evoked by the complement pathways, activated complement pathways responsible for vasodilatation, favor production, and recruitment of phagocytic cells. In this way, inflammation also responsible for innate immunity. Remember, inflammation, not only the complement pathways, even inflammation also responsible for the activation of the innate immunity. And acute phase proteins, these proteins synthesized the time of acute inflammation, and this mainly produced from the liver and also produced by endothelial cells, fibroblasts, monocytes, and adipocytes. And the best examples for this acute phase proteins are serum amyloid A, CRP, otherwise called C-reactive protein, fibrinogen, mannose binding protein, and ceruloplasmids. These acute phase proteins, which are produced during the time of acute inflammation, have got great role in the innate immunity by means of their antimicrobial activity, anti-inflammatory activity, and also they chelate the metals like iron, which are useful for the bacterial growth. And next one is the acquired immunity. This acquired immunity induced by the host immune system to a microbe, a foreign cell, or by transfer of antibodies or immunologically competent lymphocytes specific for the microbe. So this is induced by the host immune system. So the reticuloendothelial system or the host immune system, where actually the innate immunity may not participate directly, but here the host immune system has to participate and has to act against the microbe or foreign cell by transfer of antibodies or immunologically producing antibodies to produce the immunity against a specific microbial agent. So this kind of immunity is nothing but the resistance acquired by the experience of the host against the microbial agent. Yeah, there is a big correlation or interaction is there between the innate immunity and the acquired immunity. The cells which are important for the innate immunity also participate in acquired immunity. First of all, we'll see the what is the first thing is that is the what we call the innate immunity. In the innate immunity, the dendritic cell or antigen presenting cell or phagocytic cell, by means of toll-like receptors, once the pathogen entered the body, it will be taken up either by the dendritic cell or it will be taken up by the macrophage. And this one, this macrophage or dendritic cell will process it, and it may be eliminated either from the body, or if the microbial agent able to defeat the phagocytic activity, it's responsible for the setting of inflammatory process. So once the microbe wins the fight between the microbe and macrophage, then it may be responsible for the production of inflammatory cytokines, which are responsible for inflammatory reaction and also the shock. And sometimes this reaction also responsible for autoimmunity by producing autoantibodies. And if you see the adaptive immunity or acquired immunity, the relation between these two, the antigens are microbes which are which won the battle between the macrophage and the microbe. That's called phagocytosis, otherwise called the microbes which are escaped from the phagocytic mechanism will be subjected to the T lymphocyte. This T lymphocyte will be converted to the T helper cell or T helper 1 and T helper 2 cells. And this T helper 1 cell is responsible for cytotoxic activity, which attacks the cells, which attacks the viruses, bacteria, attacks the normal cells, and also attacks the tumor cells to kill them. And the T helper 2 cells are responsible for the activation of B cells to produce concerned antibodies against the microbes entered the body, and those antibodies will give protection to the individual against these microbes. So here the person is getting resistance here in the adaptive immunity or acquired immunity by exposure to the antigen. By exposure to the antigen, all these things will be taken place and responsible for bounding the immunity. And the bridging cells between the innate and acquired immunity, these cells also called as accessory cells like macrophages, dendritic cells, NK cells, complement pathway, and cytokine, that is especially the interleukin 1. These are all the accessory cells or cytokines are responsible for this kind of immunity, but specific cells of acquired immunity are T and B lymphocytes. T lymphocytes and B lymphocytes are specific cells of acquired immunity, whereas all these cells are accessory cells. All are important to initiate the or to produce the acquired immunity. And how to acquire immunity? If you see here, we have the acquired immunity of two types, active and passive acquired immunity. If you see the active acquired immunity, when the individual exposed to the microbes, either clinically or subclinical doses, or by means of immunization, or when exposure to the toxins or toxides, the host immune system will get activated and leading to the production of immunologically active B lymphocytes and sensitize the T lymphocytes. The B lymphocytes produce antibodies. The sensitization of T lymphocytes produces the varieties of cytokines from these cells. All this tries to remove the microbes from the body and to give the protection. And another thing is in case of in the acquired immunity, this another one is acquired immunity is a passive type also, where the immune system won't play any role here. The antibodies which are produced in another host would be transformed into the host or into the recipient who are in need. So this kind of passive immunity, which is also acquired, not the natural. If you see the mechanism of active immunity again, in the immune system, when it's get exposed to the antigen, it activates the B lymphocytes and sensitizes the T lymphocytes and produces the active antibodies, and T lymphocytes produces cytokines, and both are responsible for the production of memory cells. When the situation is like this, if the individual exposed to the next dose of antigen contact with the second dose of antigen, same antigen, it stimulates the memory cells and produces large number of active antibodies. That's the reason why we are giving many number of doses of vaccines to protect the individuals from the microbial agents. You see, we never give a single dose of vaccine to get protection, either a hepatitis B vaccine or polio vaccine or tetanus vaccine, whatever it may be. Minimum we have to give 3 doses or 4 doses to give the primary protection, and boosters has to be given to maintain the protection. That boosters all stimulate the memory cells which are produced during the primary vaccination, and followed by the second dose or third dose or even fourth or fifth booster doses. Large number of active antibodies are produced from the memory cells, which gives major protection. And what are the events followed by the antigenic stimulus in case of active immunity? When we challenge the immune system primarily or first time with the antigen, there will be lag for some time at the time of exposure, because the immune system has to get ready to produce the concerned antibodies against that particular antigen entered into the body. So, in early stages of this particular antigenic exposure, we can see the many number of IgM antibodies, and these IgM antibodies will be there for short period in the body, and they slowly gets decreased. And again, if you give after 3 or 4 weeks of the primary antigenic exposure, if you give the second dose of antigen, it can stimulate the memory cells which are produced in the primary antigenic exposure, and produces large number of IgG antibodies, which gives protection for the longer periods. Like this, followed by primary antigenic exposure, primary immune response will be produced with the IgM. That's a few active antibodies and few memory cells, followed by the second dose of antigen. From the memory cells, large number of active antibodies would be produced. These are considered as like IgM and IgG antibodies. And the comparison between active and passive immunity: in case of active immunity, they produced actively by the host immune system, induced either by the infection, natural infection, or by the vaccination. But in case of passive immunity, immunoglobulins received passively and acquired by mother to fetus natural route, which are of IgG variety of antibodies. Sometimes the ready-made antibodies are transferred artificially from the another host to the person who is suffering with the disease. And this active immunity is long lasting, and lag period is present, which I have shown the previous photograph, where you slide diagram, where you can see the lag followed by the antigenic exposure. So the lag period is present and memory also present in the active immunity. But in case of passive immunity, it lasts for the short time, and there won't be any lag period in this passive immunity, because the individuals are receiving antibodies, and these antibodies will be there for a short period, and there won't be any lag period here, and no memory also present. And the booster doses are useful in active immunity, and the negative phase may occur in the immune response. Negative phase means the antibodies or the lymphocytes which are present in the circulation, the total number of lymphocytes present in the circulation gets decreased for short period, followed by an primary antigenic exposure, that is called as negative phase, and not useful in immunodeficiency individuals. And in case of passive immunity, subsequent doses are less effective with the same antibody, and there won't be any negative phase in production of immune response. Soon after you give the antibodies enters into the blood and starts protection. And they are more useful in the immunodeficient individuals where the individual's immune system not working in the production of immunity. And next one is active acquired immunity. These are again of two types: so the natural active acquired immunity and artificial acquired immunity. When the people are got exposed to the subclinical doses of infection like polio virus to the tuberculous bacilli, and these low doses of the either virus or bacteria can stimulate the immune system but can't produce the disease. Because if you understand the previous concept like minimum lethal dose of the microbe, or minimum infecting dose, sorry, the minimum infecting dose of the microbial agents are required to produce disease. So here subclinical doses of infection will stimulate the immune system to produce the antibodies, but they are not able to produce the disease. And the next one is the clinically apparent diseases like smallpox, chickenpox, and mumps. In these cases, the people will get exposed to this kind of microbial agents. They stimulate the immune system, and the individual will suffer with the disease, and followed by the disease because of the activation of the immune system. The antibodies would be produced, and these antibodies will give protection from smallpox, chickenpox, or mumps in the later life. And next one is artificial active acquired immunity. Here, the artificially prepared certain preparations will be given in the form of vaccines, which can induce the immune system of the host and the produces antibodies, but they won't produce the disease. So the vaccines are the bacteria or virus are their products with intact antigenicity, but lack of pathogenicity, which can produce specific antibodies among the recipients. So, like this, the vaccines comes under artificial active acquired immunity. And the passive acquired immunity, this is the resistance that is transferred passively to a recipient without active participation of recipient's immune system. That means here the antibodies are received ready-madely. So the antibodies are prepared in some other host. From there, they would be transformed to the recipient or the individual who was in need. And this is of two types. It's natural passive acquired immunity, where the transfer of IgG and IgA from mother to fetus or baby. If the mother is infected during her pregnancy, or if you give the vaccine to the mother in the form of tetanus toxoid, the antibodies which are produced in the mother get transformed through the placenta to the baby and use protection to the newborn baby from the disease tetanus. And Ig antibodies will get transferred from the mother to the baby through the milk secretions. And next one is artificial passive acquired immunity. Here, the ready-made antibodies will be given to treat certain diseases like rabies and tetanus. Here, the rabies will be neutralized. Rabies virus will be neutralized by the anti-rabies immunoglobulins. And in case of tetanus, even though it's bacteria, the disease tetanus is produced by the Clostridium tetani, and the disease tetanus is due to the toxin produced by the Clostridium tetani. So to kill this, to denature this tetanus toxin, antibiotics are of no use. So we have to give the antibodies against the tetanus toxin. That's why the tetanus anti-serum has to be given to these individuals. Like this, even hepatitis B virus, there are so many other, even for diphtheria, we have the diphtheria immunoglobulins to treat. So here, in case of artificial passive acquired immunity, the ready-made antibodies which are available commercially in the form of sera will be given to treat certain conditions like rabies, tetanus, diphtheria, hepatitis, and so on. And the next important concept is local immunity or mucosal immunity. This is the immune response that is active at mucosal surfaces. So, by means of submucosal lymphocytes, all the mucosal surfaces will have the immunological active, and these immunological active cells are present in the mucosal surfaces of intestine, respiratory tract, and genito urinary mucosa. And these mucosal surfaces, the immunity is mediated by the secretory IgA, which are induced by the natural infection or by means of live vaccinations. And the objective of this local immunity is to prevent the microbe at the site of entry or at the site of multiplication. This needs some explanation to understand for you people. So, as I mentioned already in the topic infection, the microbe will enter into the body through certain routes and will get multiplied either in the same site or in different sites. So, for example, if you see the Haemophilus influenza virus, once it enters it through the nasal cavity, and in the oropharyngeal region it get epithelial cells, they get multiplied. Whereas if you take the polio virus will enter into the body by means of ingestion, and they get multiplied in the ileum, that small intestinal part, the ileal pairs patches of the ileum, they get multiplied. So the microorganisms will get multiplied at the site of entry, or the far away from the site of entry. So we have to fight to kill this microbial agent, not to get multiplication either at the site of entry or at the site of multiplication. This is the main concept of producing local immunity. And the live polio oral vaccine, if you give it, it can enter the intestine and in the intestinal mucosa. It produces large number of IgA antibodies in the pairs patches of the ileal mucosa, and readily the IgA would be available whenever the people consume or ingest this polio virus through contaminated food and water. They enter into the ileal part where they would like to get multiplied. Because of already the presence of IgA antibodies in the intestinal mucosa, this virus will not be allowed to get multiply, and in that way the person will get protection. So live polio oral vaccine, even though it has got some disadvantages nowadays, this is the mechanism of action. And intranasal flu vaccine against the flu virus, influenza virus, we have to give the flu vaccine that will be given intranasal route. So, if you are able to produce IgA antibodies in the nasal mucosa, soon after the entry of this virus through the nasal mucosa, they will get trapped by IgA antibodies and would be destroyed. So, this is the concept of local immunity. And next one is the head immunity, that is level of immunity in a community. So, this would be defined as overall immunity in a community towards a pathogen. And the importance of this head immunity is, if the head immunity is good in the community, the many number of will be considered as good immunity in a particular community against a particular pathogen, for which now we are trying even against the COVID-19. If everybody in the community get exposed to this COVID-19 in the lower dose, with good immunity, with good immunological functions in their body, this kind of lower doses of COVID-19 will induce the antibodies to give protection when the large number of virus entered the body. So whenever the many number of people got infected, and the many number of people have the antibodies against that particular microorganisms in their body, that type of immunity is known as head immunity. Head means a group of people got the immunity. So if head immunity is good, it will be considered as good immunity in that community for that particular pathogen. The objective of this herd immunity is to prevent the epidemic diseases and for the eradication of communicable diseases. So, for example, for the polio virus, the government of India has introduced the pulse polio program. At the same time, all the children in the entire country would be given vaccination. Because of this, the people in the community or children in the community will get the herd immunity, and the people will get the immunity against that particular polio viral infection. And the contributing factors to develop the occurrence of clinical or subclinical diseases, ongoing immunization program, and type of population and type of pathogen, all contributing factors for this herd immunity. And if the subclinical cases are more in the community, then we can have the lot of herd immunity cases. And ongoing immunization program, that's nothing but pulse polio program. That means if you give the vaccination to every individual, then the herd immunity will be developed. And type of population and type of pathogen. For example, the herd immunity will be more the people who are having good nourishment. And if the type of pathogen, if the pathogen is highly pathogenic in nature, it may cause for the death of individuals rather than providing the herd immunity. So type of pathogen also very important here. And herd immunity is possible in case of diphtheria, pertussis, measles, mumps, rubella, and polio, and smallpox. Among them, certain diseases will produce herd immunity by means of vaccination, and certain by means of subclinical exposure to the microbial agents. And these are the questionnaire that's at the end of the topic. Here I am disclosing all the answers for this particular MCQs. Just enjoy with this MCQs and thorough and prepare thoroughly. And yeah, this is the slogan which we have to adopt, everyone. Leave drugs alone, get immunity by means of vaccination, and help your community. That means understand the importance of the immunity, especially vaccination schedules for the children to avoid many number of infections in the community. This is about the immunity topic, and enjoy. And in the next video we will meet with other topic.